Hello, everyone. It's with great pleasure that I extend a warm welcome to the workshop on biomedical engineering. Today, we have the honor of hosting Dr. Yoram Vodovotz, a distinguished researcher whose studies have significantly contributed to the advance of biomedical engineering and computational biology. If you look at Dr. Vodovotz's CV, you'll be certainly very impressed, as I have been. Dr. Vodovotz is a professor of surgery, immunology, bioengineering, computational biology, clinical and translational science, and communication science and disorders in the University of Pittsburgh. Dr. Vodovotz's work is dedicated to the understanding on the mechanisms of acute inflammation, shock states, chronic inflammatory diseases, restenosis, and wounds. His profound expertise in mathematical modeling allows him to navigate to the complexities of this inflammatory condition, presenting crucial insights into their underlying biological interactions. Thank you very much, Dr. Vodovotz, for having you here this morning and also for gracing us with your expertise. And in this session, I will be with my dear friend, Federico, Dr. Federico, and also with other colleagues here from Federal University of São Paulo. So I'll give the word to you, Federico. Please feel free.
Yes, I just, well, thank you, Tatiana, of course. Thank you, Yoram, very much for accepting my invitation. Just a very quick comment. Why did I invite you? Well, of course, you know, I've been knowing you for a while. You've always been very gracious to me, and you've been involving me also in some scientific endeavors, let's say, and especially with the Society for Complexity and Acute Illness. Let's do a little bit of advertising. Well, for our students, you know, systems biology, sometimes, you know, engineering students, even biomedical engineering students may have a little bit of preconceptions. Like, why systems biology? What does it have to do with us? Well, you will see, you know, this workshop is dedicated to AI in healthcare. And we've been talking, you know, for 3 days almost about advanced data science for resolving biomedical problems. So systems biology strongly leverages a lot of these advanced computational tools, as Yoram also will show us. So this is just the reason and my brief introduction on why systems biology is really a cutting-edge frontier area for us biomedical engineers, okay? So thank you, Yoram. I will keep quiet and the floor is yours now.
Well, thank you so much for that really gracious introduction, both Tatiana and Federico. And yeah, it's a real pleasure, and I apologize that I couldn't be there in person. Just my schedule was a bit overly crowded during this time of the year. So today I'm going to talk to you about, and I've kind of given, I don't know, what could possibly be considered a provocative title on this idea that, you know, systems biology, computational biology is evolving. And also this idea that at some level you really have to be the human in the loop, you know, especially now as there's a lot of emphasis on AI and GPTs and all this kind of advances, which I think have tremendous promise. They have tremendous problems too. I think the key question is, you know, what is our role as scientists and engineers in terms of, you know, our own mindsets, how it is that we're approaching these problems. And I'm going to try to include thoughts along those lines as I present data and some of the work in our group. And of course, happy to spend time in the Q&A afterwards discussing some of those issues. So sorry, let me just, okay, there we go. Okay, I don't know, I was having this problem before. I don't know why it's not letting me. Okay, there we go. Sorry, I'm just having one minor problem with my advancing my screen. I don't know if it's a, yeah, there we go. Okay, so so first of all, I wanted to start with some acknowledgements of disclosures. First, in terms of the disclosures, I'm, of course, at the University of Pittsburgh, as was mentioned, but I'm also a co-founder and a stakeholder in a company that actually now has been merged with another company. So it's I co-founded Immunetrics about 20 years ago. Immunetrics is now a division of a company called Simulations Plus, which is a publicly traded company in the United States. Also, more recently, I took on the job of basically editor-in-chief of and founding editor-in-chief of a new journal, Frontiers in Systems Biology, and obviously some of my opinions need to be thought of in the context of that position as well. Our funding has come over the years from NIH and most recently the U.S. Department of Defense and the Defense Advanced Research Projects Agency, DARPA. The people that have really influenced me in my work are here. Tim Billiard is the chair of my department, a real pioneer of critical illness, especially in trauma. This is Eric Elster. He is now the Dean of the Uniformed Services University of the Health Sciences, which is the Department of Defense's medical school in the United States in Bethesda. And then Gary Ahn, and I think Federico knows Gary, who's a trauma surgeon. He and I have been collaborating for really about over 20 years already. And then on my own team, first I always want to thank the technical staff that have really done a lot of the work behind the scenes. Derek Barkley has just recently left us to go to another position, but really spent over 20 years doing a lot of the development of some of the multiplexing assays. That's been taken over by Jinling Yin, who's been with us again for over a decade as well. And then Phaeton Eldahabi, a really talented computational person who's done a lot of the algorithm and method development. Ruben Zamora has been with me also for pretty much the entirety of my time at the University of Pittsburgh. He directs my lab. Greg Constantine is a statistician and mathematician with whom we've collaborated really since the beginning. Chi Mi has left the university, but was instrumental in the design of the initial dynamic network discovery algorithms. And Rami Namas is a academic surgeon who also handles really a lot of the very large data sets that are generated in some of the studies that we describe. And then there are really terrific trainees. Andrew Aboud, I'll mention, was a undergraduate and medical student with us, has then gone on to medical school at the University of Pittsburgh, where you also worked with us, and then on to a residency at Mass General, and now is a fellow in cardiology at Cleveland Clinic. Lucas Shimone is a trainee surgeon who was with us and is continuing his clinical work in Germany. Nabila Jar was a computational biology grad student who's now working in industry. And most recently, Ashti Shah, a very talented medical student wrapping up her medical school career at the University of Pittsburgh with a background in machine learning, and I'll mention a bunch of the work that she has done. So in our group, Gary and I kind of coined this term back in 2008. We work on translational systems biology as applied to inflammation, and most especially is to think of it as of being applicable across a continuum of both life and health spans. So we've done work in young children and babies in the context of pediatric acute liver failure. The vast amount of our work has been in both people and animals at the kind of prime of their lives, sort of between 18 and 55, looking at various aspects of critical illness and wound healing. And then we've also done some work related to the impact of aging or association with aging. And the goal is really to unify these data across time and space that we obtain from all these different studies. And really, we learn a lot from the different studies. Rather than being only focused on one study at a time, we really often are developing methods and developing concepts for one type of study, which we then are able to rapidly apply to a different one. And that is, I think, one of the core aspects of why we've been somewhat successful over the years. And importantly, as I wrote in this editorial that I'm pointing to up here, we're talking about how to integrate the tool sets with the mindsets to try to get past this idea that there's some magic algorithm that's going to get you the answer. But what the magic algorithm is living inside of your brain. It is you with your systems understanding that then can make the right decision about how to interpret the data, how to come up with hypotheses, et cetera. And again, hopefully there'll be some time to discuss this at the end. The point is that our work really over the years, and really starting over a decade ago, was already involved on the idea of rational reprogramming of inflammation or controlling inflammation based on insights or directly by computational algorithms. And I'll talk a little bit about that as well. So as a brief sort of history lesson of our work, but also I think in general of the broader field, the central theme in our group for many years focused on systemic inflammation because, frankly, there is a complex array of processes, physiologic, pathophysiologic, going on in a human or an experimental animal, and the easiest place to interrogate is the blood, right? And so that's what we do, and in the blood you can get all kinds of side products of that, including cells, and then you can of course assay metabolites or inflammatory mediators. And the idea is that that's giving you an insight into what's going on in the body, with this assumption that sort of it's the tip of the iceberg of what's going on in the rest of the body. And that's obviously, there's many pros to this. The systemic circulation is very easily accessible. From a computational point of view, you can apply the mean field approximation and just assume that the body is a well-mixed bag of fluid and molecules just float around in equilibrium, and then which is dynamic as compartments contribute into it. But you could essentially calibrate models, for example, to the data from that, or you can use machine learning models to make pattern-oriented associations between patterns of data that you can visualize as networks, for example, and either the evolution of some pathology or the state of a patient. And for us, this culminated most recently in 2015 in a paper we published in Science Translational Medicine, where we had a sort of multi-compartment model of trauma patients. And I'll briefly allude to this in a few slides. And also from a data-driven point of view, more recently, we published on how we can use some of these data in the context of inflammatory dynamic endotypes. We've also collaborated with people like Tim Billier on single-omic and multi-omic studies, some of which have been published very recently. Now, the con of this, or the main argument against this, is there's really no direct organ-specific insight. And as I'll show you, this assumption that the blood is a direct reflection of what's going on in the rest of the body, I don't think is correct. I think it is not just an oversimplification. I think there are threshold-driven phenomena that differentiate over time and space as to when it is that you could in fact say that what you're measuring in the blood is related to what is seen in the circulation. And so if the goal is to eventually create these digital twins, as everybody is working on now, and again, from my understanding of it, a digital twin is the same as a personalized mathematical model or computational model of an individual or a system, with the extra caveat that it is being updated on a somewhat regular basis as new data come in, and forecasting of trajectories is being updated. So if the goal is to do that, and the reality is most pathology does involve multiple organs, there need to be ways of integrating across what's going on in these diverse compartments. And for us, the goal for how to do that is methodology that would involve integrating between data-driven and mechanistic models, which is an entire other thing that I've talked about and have written about. This idea that we are often siloed into the groups that are working on one or the other, but that there's now a lot of discussion of how to integrate the two. And I can just say that for well over a decade we've been writing papers that about the need to have to do this, and then moreover we've actually done work in that direction in order to help expedite that type of integration. So now we move on to where kind of we are in the lab with this idea that we can now, and again, I'm showing you a human, but more realistically, we're doing this in experimental animals. You can begin to obtain tissues from different compartments. You can begin to talk about how it is that that information in the form of various molecules that can be interrogated is either transferred or across compartments, or that there is co-regulation across compartments. And a lot of that work has been published recently. We published this initial paper in 2018 in PLOS Computational Biology, followed that up with a follow-on study in molecular medicine, and then used some new methods applied, and these two were in the context of bacterial endotoxemia, which is sort of a simplified version of sepsis. Really more acute inflammation because it's not an infectious agent, but it's often used as a simplified approximation of sepsis. And then more recently in the context of composite tissue transplantation, where you have, for example, limb transplant, and you have skin, muscle, and then you have an impact into the systemic circulation. More recently, we've begun to integrate the impact of the neural circuits into this, and we've done this in the context of both the transplantation model that I just mentioned, and in work that's currently under review also in the context of endotoxemia. We've also done it in experimental trauma hemorrhagic shock, and this was work that was, both of these two studies were spearheaded by Ashti in the group. And then finally, work in the human, which I will discuss, in collaboration with Eric Elster's team at Uniformed Services University, looking at one situation where you can get data on what's going on in a local compartment, in this case, combat wounds, and what's going on in the systemic circulation. And again, this was published fairly recently. So now let's go back to this idea of how it is we could model across compartments. So in our initial go at this, and it's the more standard way to do it, it is of course a rich field of multi-scale mechanistic mathematical modeling. You can use differential equations that represent cells in different compartments, for example, and molecules that get transferred between them. And so there's your sort of personalized model, which can then presumably serve as the basis for a digital twin. But we kind of want to be able to use data more directly into that. And so we've developed some of these, or modified some of these algorithms, including the use of dynamic Bayesian networks. We've used just a variant of principal component analysis, which we call time interval principal component analysis. Allows us to look at just basically slices of time and principal components within. We developed back in 2011 this dynamic network analysis algorithm, and more recently, as I'll talk in a little bit in more detail, the use of dynamic hypergraphs. That again is something that Ashti helped develop in the lab, and we discussed some of these ideas in a recently accepted review article that we co-helmed with Eberhard Voigt at Georgia Tech. So again, the dynamic Bayesian networks allow us to think about feedbacks within and across compartments, although they run out of steam a little bit as you add more compartments. And so this was one key motivator to explore additional methodology. The principal component analysis methodology works just like principal component analysis always does, but because we can slice it across time, it allows us to talk about what pathways predominate over any given time. And if we include data from multiple tissues, we can then sort of infer which tissue seems to be dominating or driving any multi-compartment inflammatory response. The dynamic Bayesian network analysis allows us, it does not have the feedback component of the dynamic network analysis does not have the feedback component of the dynamic Bayesian network inference algorithm. On the other hand, it allows us to look in a very granular fashion at in a very strict fashion in terms of the way the algorithm works, because it's not probabilistic, in a purely data-driven way. What can we infer about how in our case, inflammation progresses within a given compartment? And then the dynamic hypergraphs allow us to get at this idea that networks are talking to other networks. So a dynamic network in one compartment is interacting dynamically with another dynamic network in another compartment. And I think this is like a very, very important advance that is needed in order to be able to get to the idea of sort of a data-driven digital twin, which is, I think, a very important goal, right? Because the idea that you can make a mechanistic multi-scale differential equation centric model is great, but it requires a lot of thought about how it is you're modeling each process. In theory, at least, this approach with dynamic hypergraphs allows you just to directly measure the exact same things in different compartments over time, and then look to see how these things are interrelated. So again, so I'd like to break this talk up into several sections. I want to cover one area that has been, of course, a major one for our group, which is the translational applications of data-driven modeling. Again, as we're trying to integrate across, we're doing work in individual types of modeling and then coming up with ways to integrate across those approaches. So in this work that I alluded to before, this was published in 2018 and spearheaded by Ruben, this is what a time interval principal component analysis can look like. So these various graphs are looking at time courses and in different tissues. All of these tissues, spleen, gut, the heart, liver, kidney, the lung, and then the systemic circulation in terms of the plasma. And here we looked at both wild-type mice, C57 Black 6, and mice that are deficient of toll-like receptor 4 because we're giving them bacterial endotoxin, and TLR4 is the receptor for endotoxin. So here I'm only just for illustrative purposes just showing you the wild-type response. And again, this is what the data look like in their complexity. And if you look at sort of defining the ebb and flow of inflammation across these compartments by quantifying sort of, say, the top quartile of mediators, you can just get this kind of plot here. And as you can see clearly that there's almost a transfer of dominance across tissues over time. And very clearly, if you look at the plasma, which is this line here, yes, it has some features that are related to what's going on in some of the other tissues. But then there's entire things going on, like here in the heart, that the plasma clearly does not cover. And actually, this was something that was important that came out of this, which is the heart may be more than just an end target organ, but might actually be a player in propagation of cross-tissue inflammation. So we followed this study up in this paper that we published, I think in 2021, to look at what these dynamic networks look like. And this is this DYNA tool that I mentioned. Here you can see the evolutions of these networks across time intervals. Here's the quantification of them. And now I'm showing you a comparison between the wild type in red and the TLR4 knockouts in blue. And given that TLR4 is the receptor for LPS, the initial simplistic assumption is when you don't have TLR4, you don't have the receptor, you don't respond, you don't make any inflammatory mediators, everything is low. So what are we really looking at? Well, once you look at the way these networks evolve, you see a lot of very interesting non-intuitive things. Like, for example, you look over here in the lung at this particular time interval, the complexity of the networks is actually higher in the knockout mice than it is in the wild type. Similarly here in the spleen, right? And so at some level, you can think of this as a way of unifying the data across time and space, but also acting as a hypothesis-driving roadmap or a kind of GPS that says, oh, maybe you should look in the lung between 12 and 24 hours, or in the spleen between 12 and 24 hours, because interesting stuff is going on there relative to what's going on right before or right after. So this illustrates how a machine learning tool can help you to get fairly quickly to a mechanistic type hypotheses. Because of course we can also look at each one of these networks and look at the components and which molecules, or in this case, are all inflammatory mediators, which ones are interacting with each other. And so then not only can we look at the when and where, but we can also look at who and start to make some hypotheses. We took this substantially further in this more recent paper that Ashti Shah spearheaded. Now we're looking at trauma hemorrhagic shock in mice. And trauma hemorrhage is not just about a time phenomenon, it's really differential treatment. So resuscitation from hemorrhagic shock stimulates an entire set of responses that are distinct from what's happening in pure hemorrhage, because you're getting oxidative burst, et cetera, happening subsequent to ischemia. So thinking of this as a pure time evolution, like we did in the endotoxemia story that I just presented, is not really correct, because there are really different things going on, not just the same thing devolving over time. So what we did is to just sort of show this in a heat map fashion, looking across a broad array of these mediators, the same Luminex mediators that we just mentioned that we measured in the mice in the endotoxemia study. But now we're looking at as a function of the state, let's say baseline versus hemorrhage post-resuscitation, and also in the wild type versus the TLR4 knockouts. And of course, you could spend 2 hours looking through this and coming up with differences that you can find, and you can read the paper, and those are detailed to some degree. But we can also think about, okay, what is it that's going on, you know, in a perhaps more directed fashion. So here we use standard PCA, which we plot as this stacked bar graph rather than the typical 3D cube plot that people often show, because it allows us to rank order the contribution to the variance of any given inflammatory mediator, and those mediators are listed down here below. Interestingly, this suggests in the wild type or C57 Black 6 mouse that IL-17 is a pretty important mediator. And when you look at what happens in the TLR4 knockout mouse, this IL-17 moves down in importance quite a bit, which is very interesting because we've been interested in IL-17 as a very novel and relevant mediator in the response to trauma, hemorrhage, and also sepsis. And we implicate in this study, in this multi-compartmental way, that TLR4 is actually having an impact on the role of IL-17. So it's actually a very interesting use of just this fairly straightforward computational methodology. And as I said, we've implicated type 17 immunity, which is the immune cells that produce IL-17, in work that Andrew Aboud did in 2016, published in Critical Care Medicine, that for the first time demonstrating that this molecule, which is typically, the cytokine is typically associated with autoimmune phenomena, we found it to be upregulated and relevant in the self-propagation of the inflammatory response from as early as we could measure when looking at trauma non-survivors, but as compared to highly propensity matched data from survivors. So survivors that have essentially the same exact injury characteristics, same age, et cetera, have completely divergent responses from non-survivors from as early as we can assess. And this shows up in the form of the evolution of their dynamic networks. And right there at the beginning, type 17 immunity is being implicated. So this suggests this has potential therapeutic implications because one could target that pathway. There's a broad array now of anti-mediator drugs available that have been developed for the chronic inflammatory autoimmune field. And this is something that we're in discussions with our surgical colleagues. But if we go back to this data set, we can say we can do a little bit more than this. So we started exploring how it is that there is any kind of crosstalk across tissues, and then again across time and space and condition. And for this, it was our first foray into the use of hypergraphs. Now, they're not dynamic hypergraphs, these are static hypergraphs, but it's okay because this, as I mentioned, is really a problem that can be decomposed into a static one, where you can look at what's going on at baseline, what's going on during hemorrhage, and then what's going on in the process of resuscitation. And for those that are not familiar, so a standard graph or network formalism has nodes and edges that connect across. So an edge connects across two nodes. You can have weights, et cetera. In the hypergraph, the edges can connect multiple nodes. So this is essentially a flattened view of networks interacting with other networks. So if you have a network here and a network here, and you just flip it this way, now this is what this picture is showing, right? And in our work, the nodes are tissues and the edges are inflammatory mediators. And this is something that we then expanded on to become dynamic in this next study. So in any case, the point is we were able to use hypergraphs to implicate type 17 immunity and actually the interaction of multiple inferred IL-17 producing cell types across time and space. And again, I'll just, because of the amount of work involved in that, I'll just refer you to the paper for more specific conclusions and discussion. So this work led us to explore the possibility of using this, again, expanding it to a dynamic from a static formalism and trying to look to see if we could get any insights on the human situation. Again, this is work that was led by Eric Elster John Forsberg was a Navy officer working under him, and then Ruben Zamora, and then Seth Schobel pretty much runs the entire lab group of Eric Elster, what's called the Surgical Critical Care Initiative. So this was a really pretty groundbreaking study. So during the Iraq and Afghanistan conflicts, John and Eric, who were both deployed in Afghanistan, they collected clinical data and 23 serum and wound effluent inflammation biomarkers. And this is what's important. It's not just in the serum, but also in the wound effluent, so the actual wound environment. And they were assessed during surgical debridement periods, which are roughly every 3 days. And there was about 140 individuals. And you can imagine how complicated, difficult, and groundbreaking this was, because this was done during in-theater combat operations. They had various subgroups of people. We focused on 59 individuals with traumatic brain injuries, because we were very interested in this neural component, versus 81 patients with non-TBI polytrauma. And without getting into this, although you can look at the paper, there were multiple significant inflammatory mediators, but the only one that was really significant in both serum and influence and wound effluent between patients with or without traumatic brain injury was IL-17. So, and this is again continuing on the theme and making us feel that this is a very important mediator to look at. So what we then did is we carried out dynamic network analysis. And what this showed us, for example, is that in the TBI situation, you get much more complex networks of interaction. And what we're showing is the nodes in blue are in the effluent and the nodes in red are in the serum. And so we're actually showing that there is all of a sudden connectivity across tissues, which you don't see in the non-TBI case, at least at the stringency at which we deployed this particular algorithm. So if we look at that in more detail, we can look at that last time frame there. And just, again, I said you can not look not only at what and when, but you can look at who. And in this case, you can look at which mediators are interacting. The letter S stands for the serum, and the letter E stands for the effluent, so that you can differentiate between them. We've also color-coded them. And what you can see in the non-TBI case is that pretty much everything is in the serum. So it's a systemic inflammatory response. I mean, clearly there are things going on in the wound, but of the things that were measured, we don't infer some sort of self-sustaining connectivity. Whereas in the TBI setting, we actually find that this is the case. There is, in fact, a network of interaction. And as we point to here, this is wound effluent IL-17 helping to nucleate a network that involves systemic inflammation, right? And we have previously implicated IL-17 and GM-CSF as hallmarks of what are known as pathogenic Th17 cells. That was in that trauma survivor, non-survivor study that I alluded to. And we find that interaction's absolute dead center in what's going on in the plasma, suggesting that in the TBI condition, you are setting in motion a self-propagating inflammatory response that you do not see in the non-TBI condition. Using dynamic hypergraphs, we can break this story down into cross-tissue interactions. And for sake of time, I'll try to go through it very quickly. We find what this allows us to do when we compare TBI to non-TBI. It allows us to talk about what's going on within a given compartment and also across compartments in terms of cytokines that can bridge across compartments. So in this case, for example, I'm pointing to IL-17 in the serum of non-TBI patients, for example. And here it is in the serum of the TBI patients. But you'll notice that this column, which is in red, is the negative correlations or decay of the response, and the black is in positive correlations, meaning or interpreted by us to mean the propagation of the response. So the other thing that we're able to do with both DYNA and now with the dynamic hypergraphs is to talk about ramping up versus ramping down. So inflammation resolution is a very major area of study, and we can include that type of mechanism in a nominally purely data-driven modeling approach. Now, we can also find a core mediator TNF in the TBI patients being involved in the wound effluent. And interestingly, in the TBI patients, it actually ends up acting as what we consider a pro-inflammatory bridge between the serum and the effluent. So we are inferring that the ramp up of TNF is actually connecting the processes across tissues. In contrast, if you look at the non-TBI patients, here's interferon gamma in the effluent, but in these patients, interferon gamma is acting as a bridge to ramp down inflammation, which is very interesting because it's a pro-inflammatory mediator. But there have been some reports out of the University of Florida over the years that have suggested actually interferon gamma may be beneficial in trauma patients, which was controversial and confusing at the time that it came out. Our suggestion here is that, in fact, interferon gamma may be in some fashion helping to kind of coordinate a downregulatory response. And so to summarize these findings, IL-17 was the only biomarker that was elevated significantly in both serum and effluent in the patients that had TBI with polytrauma versus non-TBI, and the mediator with the most DINA connections in the TBI wards. Now, this dynamic network analysis suggested that IL-17 bridges local and systemic circulation at late time points, and then dynamic hypergraphs suggested that IL-17 upregulation in TBI patients is connected to TNF, while IL-17 downregulation was associated with interferon gamma. And again, these are associations, obviously, and we always have to be careful. And this is one of my typical comments. The correlation is not causality. But again, I think these are the kinds of modeling approaches that can drive hypotheses that can, of course, be tested. Ultimately, this continues to support our feeling that the dysregulation of Th17 responses following trauma in general, but now with this neural component from TBI, may help drive cross-compartment inflammation. And I think, again, this question of how it is that inflammation propagates across compartments, not just that it exists or that it is to track its dynamic progression within a compartment. I think this is kind of the next frontier in thinking about understanding the inflammatory response. So now let's talk a little bit about translational applications of mechanistic modeling. I'm going to actually shift gears and talk about a project that relates to COVID. And this is work that was done with Judy Day, who's now at Applied Biomath in Boston, Sujin Park, who runs the neurocritical care ICU at Columbia Presbyterian, and her fellow Ben Renard. Harinder Singh is our director of the Center for Systems Immunology at the University of Pittsburgh. And Carson Chow and I have been long-term collaborators and co-founders of Immunatrix, and Carson's at NIH. We know, of course, by now everyone knows everything there is to know about COVID-19, but remember that we started this project in February of 2020, right when this was getting started and when there was huge amounts of fear and many, many unknowns. We know that there were non-intuitive features of COVID, such as a wide range of severity and longevity, asymptomatic transmission, potential recurrence and latency of infection, and of course, the beginning to think about variants. Although, and I would argue the knowledge is still emerging, we've been in the, engaged right from the beginning in the process trying to generate vaccines and therapeutics in a very imperfect knowledge environment. And we've always said that mathematical modeling could yield insights, and I'll hope to convince you of that in the next couple of slides. So this is Carson Chow, and what Carson and I, and really it was driven by Carson who contacted me to get this started. And we did this, by the way, without any funding, without any support, and while trying to deal with the craziness of COVID and moving our labs to a purely virtual environment and all these other things that a lot of other groups were dealing with. But our thought was that most mechanistic models and most of the thought process in the immunology community was that this was something like HIV or more likely influenza. So a respiratory virus that maybe also can be a little bit bad. And of course, influenza can be quite bad. And we reasoned rather that the responses to SARS-CoV-2 shared many features with sepsis and critical illness, and that this was being missed at the time. And then, of course, some of this work was starting to come out and get into the popular press that you might be in a sepsis-like phenotype. We thought we would leverage an extensive background of mechanistic mathematical modeling, including early work that was our seminal paper in 2004 that Carson spearheaded on simplified mathematical models of the inflammatory response in critical illness towards COVID-19. And if you look at that work back in 2004, already then this predicted that under different model parameters, a really simple model that just had the bacterial pathogen, generic inflammatory cell and what we would call a late mediator, which is really damage-associated molecular pattern molecules or DAMPs, could in fact be sufficient to encompass a wide array of different patient responses. And we could demonstrate, if you look at this, everything from a recovery to the infection, low-grade, ongoing, repeated infection, persistent infectious death, or immune failure or sterile death. And if you take away recovery, that leaves you with 4 basic phenotypes of patients that are presumably doing badly. And it's really quite interesting because I had the privilege of participating in this very large multi-author study that was published in JAMA that identified, guess what, 4 endotypes of sepsis. And our mathematical model that preceded that by 15 years predicted that essentially. Now, one of the core features that we argued was operant in COVID was not the intuitive idea that you're looking at typical type 1 interferon responses, because already early on it was known that these are actually impaired. And yet, how can you imagine that this inflammatory response ensues if the virus is impairing the primary mechanism that is inducing that inflammation? So we reasoned that this maybe was unmasking DAMP signaling, meaning signaling to damage-associated molecular patterns which are released when cells are stressed or damaged, secondary to the viral infection. And this was the structure of our mathematical model. Basically, we had a 4-compartment model that had virus-infected cells that become damaged and release DAMPs. That stimulates an innate immune response, which is necessary to drive an adaptive immune response of, say, T cells and B cells. But that there is this other mechanism wherein, number one, there's cross-regulation between adaptive and innate immunity. Also, you can get into this self-feedback mode where you're having damage to inflammation to more damage, which has been a core feedback loop we've talked about for many, many years. And when this ramps up, it actually suppresses adaptive immunity, creating a situation of sustained inflammation simultaneous with suppressed beneficial adaptive immune responses to the virus. And again, all of this implies a central role for DAMPs. And it's secondary to this attenuated PAMP response, as I said, which is needed. So you need this loop to eliminate the virus, because we reasoned you have to go through this DAMP cycle to get to adaptive immunity so that you can eliminate the virus. But the risk you run is, under the wrong set of circumstances, you are left with this potential persistent inflammation. And also because you are suppressing innate immune, overactivated innate immunity can suppress adaptive immunity, you can allow for persistence of virus-infected cells. So it was 4 differential equations. And really, we made the world's most simple assumption that there was a single virus-infected cell as a starting point, even though you can't detect that. But that we assumed that everything after that is all about inter-individual differences. So we're not assuming that there's a virus load-dependent difference in outcomes. It's really just about the host response. And that shows in this picture of the 4 archetypes that we came up with, which are recovery, again, like our sepsis model. But we said that could include a long ICU stay. And importantly, this has a prediction of an asymptomatic period, which I'll talk to you about in a second. Uncontrolled infection, this recrudescence or regrowth of the virus or long-lasting disease, and then damage, sustained inflammation. As I said, there's a period that's inferred wherein early on you are having virus, but you're not having any symptoms because you don't have sufficient inflammatory response activation because of the fact that there's this time period of delay until the DAMPs begin to appear and drive inflammatory activation. So this is essentially a way of thinking about asymptomatic infection. And this is where we stood. We were at this point in August 2020. Now, I should note that later studies have shown, as this one here in Cell, after getting a lot of data and a lot of time, that there are 4 endotypes. We predicted 4 archetypes of this response to COVID a couple of years before, using relatively little data. So we did have data, right? We had data on the patients, which is covered in the paper that we published, that I won't have time to go into directly. Now, we also simulated vaccination and suggested that vaccination would help all of the 4 archetypes of patients. Again, for reasons of time, I won't cover it. But more recently, we pursued another prediction of the model, which is that there's this potential for Paxlovid rebound, meaning that Paxlovid, the drug, which is an oral medication, antiviral, targeting SARS-CoV-2, might lead to bounce back. Now, how does this occur? Well, initial studies, the ones that led to, and that were part of the clinical trials, were performed in unvaccinated patients, meaning, of course, you have a virus, you don't have a pre-existing immune response to the virus, you need to kill the virus, and it helps you, okay? However, most individuals have now been either vaccinated or experienced SARS-CoV-2 infection. What does that mean? That means that you actually have a pre-existing response, and now you're starting to get this idea of a Paxlovid rebound, and we hypothesized that these two issues were linked, right? And this shows up in this kind of a simulation. Basically, if you are unvaccinated, what will happen is, so here's the minus vaccine. So this would be the base control case. You're getting this much virus, right? If you are not vaccinated, you give the Paxlovid. Of course, you reduce the virus, reduces everything. Is very nice. Now, if you are a vaccinated individual, which I said before, our model, this is the same model I've been describing, it suggests you will do a good job of suppressing the virus. That's all good. However, if you've been vaccinated, right, the reason that you begin or are prescribed Paxlovid is because you exhibit symptoms. But we suggest that the symptoms are due to the fact that your pre-existing immune memory that you got from the vaccine stimulates that initial cytokine production and fever, etc. Now you get Paxlovid, and yeah, it works, but the problem is you would have been working regardless. And the problem is you then take away the one trigger for the rest of the immune response because you give the drug, and now the virus comes back up because it's logistic and it doesn't really ever get fully killed. And this is the bounce back. And we can demonstrate that in this kind of schematic that I've just said. Really, it's a question of timing. When do you get the drug relative to the onset of symptoms? And also, what are the symptoms due to? In an unvaccinated person, the symptoms are due to initial innate immune activation. In the case of a vaccinated person, they're actually driven in a circuit that starts with your B cells and T cells being recruited, which then secondarily leads to innate immune activation, fever, etc. So this is actually a very interesting use of the mathematical modeling approach that I've just described. But more importantly, and these are the core features that we came up with, just to recap them, that we have a parsimonious model that's based on this non-intuitive idea that when you inhibit PAMPs and type 1 interferons, you're actually unmasking a secondary DAMP mechanism, which is just a bit late and not enough. And so it allows the virus to propagate. And this mechanistic model could be used to predict effective therapy. And again, as I'm not showing, is simulated vaccination. And the prediction of this Paxlovid rebound. And I should mention that we've also been collaborating with Sujin and Ben on some more data-driven endotype type work in COVID as well. But we can think about this and the implications in a more large-scale fashion. So maybe we should be rethinking about what viral infection is about. So if you think about a kind of continuum of how much damage is induced by viral infection of cells, you could imagine that at the low end, for a given amount of viral infection, you get only a little bit of cell stress and damage, which would be maybe what happens in flu or the common cold. And at the other end, you get a huge amount of damage. And by the way, it's an optical illusion for those that can't see it. The CV is the same size in both cases, just the damage is really big. But now that's maybe characterized as SARS and MERS and Ebola and Marburg, all these viruses that are massively hemorrhagic, necrotic, et cetera, and cause big stresses. And maybe SARS-CoV-2 is living somewhere in the middle. And we could argue that the variants are driving you more in one direction versus the other. Delta variant, if you recall, had associated with worse disease, et cetera, worse symptoms, suggesting that maybe it's making you have more DAMP release. That's a hypothesis to be tested. Whereas Omicron and the Omicron variants we're dealing with now are probably aiming more in this direction. I would argue evolution drives you in this direction so that the virus can persist, because this is self-limiting. You basically kill the individual, and therefore the entire life cycle has to start all over again with an opportunistic infection at some other location, as what happens with Ebola or Marburg. And as I said, we had data on some of these things. So just one of the core phenotypes, which is this, what we would call the long COVID phenotype or archetype of a patient. This was from our model. This is data from an exemplar patient from Columbia Presbyterian that Sue Jin's team had, and this is PCR test. And you can see that there's an initial elevation in this patient. Then there's disappearance, or initial positivity, then there's disappearance, and then there's recurrence of positivity, and there's disappearance. And this person stayed in the hospital for many, many days, right? And it looks a whole lot like what our predicted simulation, which was not calibrated to data from people. Again, it was actually calibrated only to some initial data from monkeys about roughly how much or how soon virus peaks occur. Now, what's interesting about this is something that everybody thinks of for COVID, but now what I've just said to you here is that this is a feature that should be accompanying every viral disease. And in fact, there's a recent study that's just come out that suggests that you get these long-term symptom profiles in very many other acute respiratory infections, which again supports the idea that this is a general feature as implied by the mathematical modeling that we've done. So, we've also used this in larger this type of approach in larger models of inflammation, as I've alluded to, this kind of differential equation-based models. And this is something that in my role at the Frontiers in Systems Biology, I've tried to promote. Gary On and others are. Gary On is a section chief editor or specialty chief editor in the journal. We've had special topics on this topic. Just really briefly, this is the way we did it originally. We did a 2-compartment model of inflammation in swine, which were then expanded to a 3-compartment model that became the basis of this paper in Science Translational Medicine back in 2015. More recently, we expanded this to think more about how hemorrhagic shock evolves and coagulopathy in these patients. So taking this, and this will work with Immunetrics and a wonderful collaboration with Jeremy Cannon, who's a trauma surgeon, actually also at military, but has been at Penn at University of Pennsylvania in Philadelphia for quite some years. And we took this model that I've just shown you, but now expanded it to include aspects of the coagulation pathway, and then obtain data. This is data from collaborators who were involved in what was called the PROMPT study, which was a study of various transfusion approaches for shock hemorrhage, and also working on the idea of the core goal was to think about how strategies such as lyophilized plasma might work. And this was work that was done by Marty Shriver's group, and their team was part of this and had access to the animal data from that. And that was really one of the other goals of this. And on a more philosophical basis, the goal was the possibility of integrating clinical with preclinical data into a single workflow using mechanistic mathematical models. And this gives you a sense of the kind of things that we did. And this paper is currently under review. But a combination of preclinical and clinical experiments and showing you the differences. I think, as everybody knows, in preclinical models, you can get a lot more data. You can interrogate more, you can look at multiple compartments, et cetera. But you're usually limited by the amount of time. You typically have to euthanize the animal after a few hours. Whereas in the clinical setting, we typically don't have that early time point data, but we have the longer term outcome and, of course, the relevance to the human disease. And we combine that with in silico experiments in both porcine and human subjects, where we can think about different resuscitation modalities and their impact. Now, this is a very big study, and again, for reasons of time, I'm just going to enumerate some of the core outcomes in this kind of bullet point format. We had accurate prediction of physiologic, inflammatory, and laboratory measures in both swine and patients. We could predict the outcome and time of death for patients in the PROMPT cohort. We suggested, based on simulation, that resuscitation with plasma and red blood cells together could outperform resuscitation with crystalloid or plasma alone. And again, for people that are not specialists, we can get into details of what these things mean. But I'm just trying to get across the core translational power of this type of approach. We also suggested that earlier plasma resuscitation could reduce both morbidity and mortality. And this really harkens back to a paper that Gary and I, along with John Bartels of Immunetrics, wrote well over a decade ago, suggesting that mathematical models could in fact bridge the drug development pipeline from preclinical to clinical studies. And we're trying to use this as a kind of example, not just with specific applications to trauma hemorrhage, but maybe broader applications in the development of novel therapies for any disease. Finally, we're talking about this idea of integration. And so how are we actually integrating? This is sort of the workflow that we've used. The idea being that we can get data, high throughput series of mediators, infer causal relationships using some of the data-driven tools I've just been describing, pull out key features from that, and now encode them as mechanistic models, and then map these to patient phenotypes. And this allows us for what we might call model-based precision medicine, this whole digital twins approach, which would require further clinical validation and set up this kind of a cycle. But we also want to get at this point of how can we rationally reprogram? And for that, we can make use of the field of dynamic-based or model-based dynamic control, where you could actually actuate control. And this is something that I'm sure it's, you know, that you in bioengineering have had some exposure to. And some examples of this, we've actually worked in the past to generate an adaptive negative feedback circuit to actually reprogram inflammation. We published the original study on this in 2012, and this was the most recent follow-up study that was recently published on this, and happy to discuss it. But where this has manifested in a really large way is in this DARPA-funded, more than $20 million project spearheaded by Steve Badalak here at the University of Pittsburgh, Sahi Cohen-Carney at Carnegie Mellon University, and myself. And the goal is to take some of the computational work, some of which I've not described, but is related to tissue-realistic computational models of wounds. And this is work that we did jointly with Gary. And Gary, who's spearheading the augmentation of these initial models into what is effectively the world's most sophisticated agent-based model, three-dimensional model that runs on a high-performance computing cluster of a wound. Because we're carrying out experimental and also human data analysis of complex volumetric muscle loss injury wounds, which are combat relevant, very different from the types of diabetic wounds and other wounds that a lot of groups study. Steve's lab has generated a canine model of this, and we've published some initial papers on this in terms of data elements that we get, everything from histology, protein level analysis in my lab, transcriptomic analysis, et cetera. And the goal, and this is that paper, and the goal is to really have this virtuous cycle between machine learning data-driven models to infer core features and modification of this WABM, the wound environment agent-based model, and prediction, and importantly, deep reinforcement learning-based control, so that we can actuate the control on the wound. And then the idea is not to have this needing access to a supercomputer, but to encode the core control policy into firmware that will then sit on this electronic device that has sense and actuate capabilities that is being spearheaded by Tsahi's team. So finally, I'll just leave with this, what I would call an aspirational or translational vision for systems biology. And we can put it in critical illness, or you can think of it as really any disease. In our case, work that we've been doing in large collaborative teams, we do broad sort of large scale omics analyses from patients, and we've published on some of this. Some of that has been spearheaded by Tim Billier. That's combined with, and I haven't talked about this technology, but I think people may be aware of it, sort of single cell immune omic type analyses that are deep. And again, we've had the privilege of participating in some of these kinds of studies. The goal is to integrate these data into predictive models. But then what are those models being used for? Well, they're being used for potentially patient stratification and novel insights. But ultimately, we really want to use them for rational reprogramming, examples of which I've tried to show you. And so I'll stop there and just thank the much broader team, and then maybe I'll stop sharing my screen and hopefully have some time for questions. Thank you.
So thank you, Yoram. Of course, an incredible talk. Thank you so much, really. As you can imagine, I'm quite familiar with at least some of the things that you presented. Before opening the discussion, I would just like to make some comments, especially for our students, again, along the same lines of what I commented at the beginning, that even though, maybe we don't know a lot about some biological details, pay attention to the fact that what Dr. Vodovotz presented, and a lot of research, not just in this field, as you said, this rationale is not exclusive to critical care, acute illness. You can apply the same concepts to neuroscience problems, for example, to organ transplantation, and so on and so forth. But maybe you noticed there are lots of concepts from bioinformatics, from control theory, circuits. So, courses that you already took in your undergrad, really, it is very close. A lot of these tools, these methods are very close to what you study almost on a daily basis. Heat maps, at the end, you see, this is all meant, for example, as in the final example that Dr. Vodovotz showed us, to create the devices. So it's all about making circuits like you learn to do in a lab setting. So this is just, again, to complete this continuum, this demonstration of the continuum that there has to be between biology, disease, and engineering. At the end of the day, as bioengineers, we should aim to provide either decision support systems that doctors can use, or even patients can use, or devices to contribute to solving disease. Anyway, I'll stop.
Yeah, no, I wholeheartedly agree and support. I think that what goes along with that isn't automatically stated and is possibly missed sometimes in the need to educate students in the actual technological aspects of what they will need to do, is the conceptual aspect of this, right? As I try to get across and to allude to, there is no magic tool set. There is, however, a mindset. There is the way you are thinking. And the problem is that the typical classes, which again, I'm not blaming anyone, the typical classes have a hard time of actually getting that idea across. Mm-hmm. You're sort of supposed to kind of figure it out from everything you're taught. But because especially, and I'm especially concerned about the prominent role that pure machine learning is having now, there is the potential for an unspoken message, which is, hey, your brain doesn't matter. In fact, your brain is going to cause errors. Let the computer tell you what to do. There you have the black box models. You have, because, massive neural nets, etc. So this whole push now for things like interpretable AI, well, another way of thinking about interpretable AI is a mechanistic mathematical model, and because it's inherently interpretable. More importantly is the human who is either thinking about what to include in the model, because you have to have a certain understanding of the process in your head if you are to actually encode it as a model, right? Or even more, I would argue, you have to have that understanding in your head if you are to interpret the results of a machine learning analysis. Because if you're encoding it as a model, well, okay, you have ideas, you can have a debate, and you can say, well, I think it's this and this, and you can put things on a board, and you can decide, and then there's trade-offs, and then the scale, and you can make it happen. Okay, you're a direct participant in that process. But when you have the machine learning output, boom, here it is. Okay, first you have to understand what are the parameters, stringencies, p-values, et cetera, that lie underneath that. But once you put that aside, you go, okay, great. Now what does it mean? Oh, I don't know. I mean, I see this and it's there and there's a thing there and there's maybe, I don't know, right? And we've been in the situation where people have come to us and said, we've done all this informatics analysis and we have no idea, right? So then they talk to the informatics experts who then said, hey, listen, I don't know the biology. I just know the informatics. It's up to you to figure out what's going on. And so there's this valley of death in the middle where somebody has to bridge that gap. And to bridge that gap, you have to know the underlying biology and physiology in medicine, and you have to understand the strengths and weaknesses of what's coming out of these models. You have to understand, have some intuition about the disease so that you can say, oh, interesting. Over here, I see this connection to this and this, and it's kind of central, or maybe it's not so central. So maybe there's this other thing over here, but look, I already know that there's some literature about this or this, right? Some intuition about what's going on, meaning you have to be able, it is almost more imperative now to know the knowledge about the disease or the biology than it was before all this computational modeling was apparent. And of course, a lot of the reason for driving all this AI is to make it so that people didn't have to know as much, right? But actually, I think you now need to know more, not less.
No, absolutely. Before giving the floor also to Tatiana and the questions that she typed in here, let me just tell you this. We just had a 2-hour roundtable to open the morning, and we had 3 experts in AI. And we actually touched upon on some of the issues that you raised. So it's actually great, for our audience that you also came to this. And well, first of all, general comment in our area of research, critical care. Sometimes people come to me actually, I was last month at European Shock Society meeting, which is mostly basic scientists. I mean, I've been involved with them for a while, but it's funny because, there was me and another engineer, and it's like, oh, you're engineer, so you do AI. And I'm like, no, I don't. It's honestly not my field as an engineer. I do mostly actually hypothesis-driven modeling, okay, mechanistic modeling. But other than that, and actually there was some medical doctors that had presentations on endotyping. Billier actually presented there, Dr. Billier. Yes. We don't even know exactly what sepsis. That's why, all these endotyping featuring and stratification that you showed, for instance, from your studies and others, help in this regard. We have a definition that's a consensus definition. It's set in stone until sepsis 4, right? Which is based on symptoms. So do we really feel it will be enough to just introduce more advanced models? Okay, so comment number 1. Comment number 2 that came from this roundtable this morning. So one of the take-home messages of our guests was, honestly, there's plenty of data around. So it's not like I want more data and then I will do something with them, because now we have very powerful machines, the hardware, and very powerful software. Okay, what's the question? And as researchers and as educators, and actually maybe some colleague who's here with me may chime in before you go in a few minutes if they want. So, we have to rediscuss what type of biomedical engineers we're educating. And maybe on your side, on your end, since you work in a medical school, medical doctors or, medical researchers in general, immunologists, pathologists, whatever. Because things are changing, but it's just an illusion thinking that we just have more. Well, we want to become lazier. I understand the society has been moving at all levels towards this. But, if we don't have the right questions, we either build insufficient models, or we can't interpret what we get out of our fancy bioinformatics pipelines and tools and this and that, and we end up.
Well, even more importantly is this issue of scalability. I mean, those are all gigantic questions you just raised, and we can definitely discuss that. But even more importantly is the fact that it's impossible to scale. So the purely data-driven approach means, in theory, if you stop in absurdo reductio, if you stop and think about it, it means you will need to make a data-driven model, multi-compartment model of data being obtained at the omics scale from every person on the planet for their entire life, while also treating every one of those people with every drug there is and getting that same amount of data, so that later you can figure out which is the thing that works. Okay, so I think I've just described a thing that is completely impossible, right? Not to mention incredibly expensive. Right? Yes. The alternative is principles-based mechanistic models that are good enough to reproduce the virtual behavior of populations, that are good enough to reproduce the core aspects of trajectories of individuals, and can talk about in simulation with the impact of different therapies. And that tell you that you only need a few measurables to be able to decide which one of these to classify you, rather than I can only classify you when I have this gigantic data set, which is already bad enough. But then to figure out what to do to you, I have to create that same size or bigger data set for that plus therapy. Right?
Insane.
So I literally had this debate with the person who at the time was running the IBM Watson program at an NIH meeting in like 2008. And, you know, Watson was running high and everything was great and whatever. I predicted those guys were going to go out of business. And, you know, they went out of business because it wasn't scalable. They were spending more money within IBM to seed, to get people to use that platform so that they could demonstrate that the platform was useful. It became the typical tail wagging the dog scenario, right? So then, of course, they ran out of steam and left it. And frankly, similar stuff is going on with the large language models now. As long as somebody can put billions of dollars into something, and then you can create a chatbot that sure seems like it can talk to you and whatever, it's all good. But then you check, and it will give you any answer you want. It's almost like this dog that really wants to please you, and will just bring you stuff, right? Sometimes it brings you stuff you want. Sometimes it brings you stuff you don't really want.
You know, Yoram,
Yeah.
this morning, one of our guests, I don't know if you know about that because it's a case in the US, he said that he's based in the UK, so he was reading this morning there, that there was a court case in the US where an attorney brought up, they sued probably an airline. Do you know about that?
Yeah.
I mean, we were shocked. We didn't know about that. That's insane. So ChatGPT even created a case. So imagine for us, you know, we talk about, and I'm warning against that. When I say fabricated data or fabricated papers, I'm not talking about, you know, throwing directly under the bus the whole idea of virtual clinical trials, because for that we need mechanistic models. I'm just saying, let's not confuse the two things. But let's be careful, because it might create a word that doesn't exist and say, oh, here are the data. I simulated them. These are, consider them like surrogate data, whatever. But actually, you know, that's a physics that don't exist.
Exactly.
Like,
well, so I mean, so you notice I'm being very, number one, I don't want to fall into that trap. I don't want my group to be working on stuff that is just because it's trendy. I want my group to be doing things that help provide knowledge rather than blind alleys. And so you notice that when I mentioned, so first we realized we needed dynamic data-driven models when everybody else was just getting more and more and more data on single time points. Okay. The reason we did that is because we were already appreciating the need for dynamics in the mechanistic modeling side. Second, we focused at the protein level rather than the transcriptome level because frankly, you know, like they say, talk is cheap, mRNA generation is cheap. So a cell will generate mRNA for everything it possibly might have to deal with, and then it'll begin to focus itself at the protein level and ultimately the metabolite level as it gets substantial enough information to tell it, this is the thing that's really going on. The analogy that I use is a boxer, a tennis player, a basketball player. You have to move around, right? So because if you stand still, it's less energy, but then the ball can go here or the punch can go here. And so you really need to be somewhere here. Okay. But so moving around turns out to be a good strategy. Now, if you move around so much, you use all the energy. Now, not a good strategy because you're moving so much around, the ball goes over here, the ball goes over there, you're too tired to get to it. Or you jump so far over this way that when the ball goes over this way, you miss it. Okay. Or you move your head right here just as the punch punches you in the face. So the point is that there is a trade-off of how much moving around you do. Okay. So I think the cell is evolved to make a lot of RNAs for the possibilities that could happen. And then it gets more information. And then it says, okay, there's a natural decay rate to the RNA. So that means the fact that I generated a whole bunch more options than the ones I need, I don't care. They'll decay. I don't have to spend a ton of energy to make them go away, right? And now I'm going to begin to make some proteins. So we focused on proteins when everybody else was doing transcriptomics because, and then they were finding, guess what? The signature of everything is everything. They were finding that stress is the signature of everything. Well, that's a giant surprise. You know, that's crazy. Who would have known? And then it turns out you didn't know anything more than you did before. You just knew it to a, you were wrong, but now instead of being wrong to 1 decimal place, you were now wrong to 12 decimal places, which is still wrong. Okay, it's 1 plus 1. If you say that 1 plus 1 equals 5 or 5.0 or 5.00000, it's still wrong. But it makes you feel that you're right because look at all these decimal places that I got. Well, that's the measurement. That's the fallacy of chasing after just more data for more data's sake. So the idea was you have to spend time thinking about what is actually going on, and then and if you say, okay, based on what's going on, I need to either use or generate tools and approaches coupled to the way experiments or clinical observation is done so that I can get the answers that I want. Again, when everybody else was doing clinical studies trying to recruit thousands of patients with getting 1 data point, and that data point was usually late, let's say at least 24 hours into critical illness, for example, we reasoned we needed multiple data points and earlier.
Yeah.
So we focused on trying to get 3 data points in the first 24 hours. This was in a big NIH Center grant funded study. The fact that we did that generated decades worth of papers afterwards and insights and findings because we did the study right. We focused on protein level data and we focused on multiple time points in the first 24 hours and then daily sampling for a week. That alone has led to everything, including a lot of things we never predicted we would find, like novel single nucleotide polymorphisms associated with mortality, the ability to do propensity matching, the ability to capture the broad heterogeneity of trauma while having subgroups of patients of large enough size to get what we want, the acute dynamics of what's going on, the fact that the current state of critical illness is no longer what it was at the start of the study because protocolized care improved it. And actually, it turned out the signal is mostly there in those early time points. So everybody that went to 24 hours, the majority of the action was gone and they weren't seeing anything. I can enumerate, again, it's not to tap ourselves on the back, like say, look, I'm a genius. It's just to say it's common sense. The piece that was missing from these other approaches is to stop and think, is the emperor naked or is the emperor wearing clothes? And if you tell yourself, no, look at those beautiful clothes, right? Okay, well, maybe. But if that's what's going on a lot in these fields is that you end up with a lot of people who get together in a group, in a consensus. Every one of them is affirming the fact that the emperor is wearing wonderful clothes. There's a reason we get told those stories when we're kids is because actually the human behavior hasn't really ever changed, right? It's just people forget to remember those stories and then they don't realize they apply to their life later on as well. In any case, that's just my little rant, but I apologize.
Oh, that's true. I mean, you also made me think one of our other invited speakers showed us the Stanford definitions of AI, which, how rational, how human you want your system to be, which then conflicts with our limited rationality, but at the same time, how about the ethics of an ideally completely rational system that maybe will take decisions? What's going on with me? I don't know.
Yeah, you look cool.
Exactly. Maybe we'll bring to the side, we have a patient that's gonna die anyway in 1 year from now. We're down to palliative care. Let's get rid of them right now because cost is, the criterion for success of our model. Hey, Tachi, do you want to, bring up the...
I know you have some questions. Probably, I apologize. I want to make sure there's enough time.
No, it's okay. Thank you, Dr. Vodovotz, for this fantastic presentation, for this lecture. I have one question. While you were talking with Federico, I was thinking about the some markers that we use in some disease that has a biphasic or a dual response. I mean, in some stages of disease, talking about chronic disease, or even in acute disease, they go up and then they go down, and in some cases they go up again. So how difficult is to address this using these mathematical models, or how do you address these issues? Sorry about this, and how about the combination of different markers? In this case, we cannot exclude them, this is very difficult to track the behavior of these markers, but in some cases they are essential for the understanding of the disease. And how do you track them?
So I'll start with your second question. So one of our first forays into integrating data-driven and mechanistic modeling, and by the way, I apologize that if I had had more time, I would have covered another study that we published recently that really directly demonstrates how to do this integration that I showed as a concept. But the very first way we did it is to measure a large array of mediators at the protein level, and then to do PCA as an initial data reduction, and then take what came out of that and use that into a framework of mechanistic modeling. So basically the idea was they're not directly integrating, but you're using something like PCA to tell you where's the signal in your data, and then you only focus on the part that has the core signal. Because at the end of the day, a model is an approximation, and you want to not just throw in your favorite mediator because you've spent your whole life studying it, but the one that the data actually tell you is the one that you should look at. And that's actually another part of this. Modeling is objectivity. Modeling is about truth. I mean, actually, your brain model is about truth too, right? Because if you are deluding yourself about what's going on, because you're creating a mental model in your mind that tells you whatever, the earth is flat or the gravity doesn't work, well, you're going to step out of a window and you're going to find out, well, gravity does work. So it turns out you made a mistake in your mental model. So modeling is about objectivity, and successful modeling is about getting to the truth. And so that means that you need to set it up so that you are able to give yourself a chance to get to the truth. So in the case of this, yeah, you measure many things that you believe to be relevant, not just everything there is. But even if you measured a bunch of things that are not relevant, something like PCA will rule them out pretty quickly. And so then and there's better approaches probably that you can use, but I'm just saying a simplistic way. So you do that, and then you take the rest of that and you encode it into mechanistic models. And we did that in, for example, in the case of traumatic brain injury in a study that we published in 2016. So that's one question. And the other question was about the multiphasic time courses of things. So to me, first of all, when you see the multiphasic time courses of things, your brain should instantly say, this is something that you need to model mechanistically using a set of differential equations and set of feedbacks that are able to do that, right? Because now that doesn't mean that you can't intercorrelate dynamically those kind of time courses, and that's what we do. The dynamic network analysis that I showed is fed by time courses of data that look just like that, multiphasic. And again, depending on the stringencies and some trade-offs that you employ, you find that in fact, yeah, things can cross-correlate, and then the dynamic evolution of the networks often has that same kind of multiphasic behavior. So to me, it's not a problem. It's not a bug. It's a feature. You know, it's the inherent nonlinearities of the system that should tell you that it's not going to be a simple answer like IL-6. Okay, it's not going to be oh yeah, just measure IL-6 and you're fine. Okay, which is a thing we've been railing against for many years. In fact, a core piece of that Science Translational Medicine paper was to hit IL-6, the sacred cow, kind of fairly directly, to show that linearly every single piece pointed to IL-6, but when you put it together in the model, it's completely not at all about IL-6. And we showed it. And I think that's what the editors of the journal really liked, because it highlighted the way it is that people delude themselves, and how actually some elements of the data and data analysis can totally lead you to that in isolation. But when you put them together and you objectively allow it to tell you a story and a decision, and you follow that advice, it actually kind of takes you in the right direction. So again, I'm not trying to say that mechanistic modeling is the end all and be all. I will just use the example of what we did during COVID. The whole point was a world that is based on gigantic data streams and machine learning has a problem when a disease is brand new, shows up, and there are no data. What do? What's your solution to that problem? Oh, I guess my solution is to lie in a corner and die, I guess, because the only way I can live is to get enough data. It's going to be extra special if a whole lot of people die, some of whom are going to be the ones getting you the data, in which case you will, again, never get the data. So that means you have set yourself up for extinction, which seems like not a good idea, in my opinion. So instead, we said, what could we measure today? What could a physician or a health worker measure today? They could measure 4 things. They could do PCR tests, even though there were hiccups early on, especially major mistakes made at the CDC labs that were frankly amateur. I mean, there's people in my lab that could do a better job of creating PCR tests. But in any case, leave that aside for a moment. Fairly soon, they corrected those errors and there were PCR tests. So you could measure essentially a marker of virus infected cells. 2, you could measure cells if you needed to, or you could certainly measure inflammatory mediators from cells, because we said there are no magic inflammatory mediators. They're the same mediators, and in fact, everybody was looking at the same ones, IL-6 and TNF and whatever. You could measure the mediators, you could measure antibodies, which they could do eventually, right? And if you really wanted to take a little time at single-cell RNA-seq level, you could, of course, measure cells, or you could just do flow cytometry, right? The bottom line is you could measure every one of the things that we said you should measure pretty early on following the induction or the understanding that this was a worldwide pandemic. With that alone, we said you could calibrate these models and you could segregate individuals. And you could say this person lives in this space and this person, and that's what we showed in the paper. Again, for reasons of time, I didn't show it. But we had exemplars of every one of these non-intuitive behaviors. There were questions early on about, transplant patients are on immunosuppressants. They will probably be incredibly susceptible. Our model predicted that actually it's the other way around. You're going to linger for a long time in the ICU, but you're going to do just fine. They had data on this, and then it started to get published. Hey, our transplant patients are not doing so badly. What's going on? We go, okay, figure 3. So, this was actually a very frustrating thing for us, because we literally, at the pace we were working, we were almost literally either saying something and then a news item would come out, or a paper would come out, or an archive, preprint would come out within a few days. And we kept adding more stuff because we wanted to fully solve it, but also because the pace at which everything was developing was such that we felt that we needed to, we had an opportunity to integrate everything. So, but again, it points to the idea that you absolutely can use reduced mathematical mechanistic models when you know very little, because you know something. You never know nothing. If a microbe came from outer space tomorrow, we still would know how our body responds, because our body isn't going to change. Yeah.
Yoram, if you have one more minute, a student here brought me a question that she has for you.
Okay.
Natalia is her name.
Betania.
So she's Betania. She's asking whether the type of models that you presented could be extended to model inflammation, for example, in the case of autoimmune diseases like multiple sclerosis, or this is the part that I add, probably they're interested in chronic disease in general.
So first of all, there are many groups out there doing that. And what I would add to that is that, so first of all, it's being done often behind the corporate firewall, because that's where the real market is in chronic diseases, not in the acute diseases that occupy our time. However, I would also argue that the acute diseases, and especially trauma, because you have a situation where you're generally otherwise healthy, and then you have a severe event, and then poof, everything shows up. So you can see times 0, and it gives you an opportunity. Again, we happen to have that opportunity with SARS-CoV-2, but only because Sujin's team and at the hospital system, they had people in the hospital who became positive in the hospital and were being monitored. Otherwise, you just have to wait until someone shows up, which is what happens with sepsis as well. Anyway, that's a separate issue. But the point is that a lot of the insights that we've obtained from trauma, like this IL-17 story that I was referring to, suggested that the mechanisms evolve as early as you can measure once you have a severe enough stress. And those are the mechanisms that are hallmarks of autoimmunity and chronic inflammation. So the idea is that actually there is no new wiring. There's just, now we can see what the beginning looks like. Whereas in typical chronic inflammatory disease, you just don't know what the beginning looked like. Was it a viral infection when the person was a child? Was it a head injury when they were an adolescent? What was it that triggered that autoimmune reaction? A genetic predisposition that just chronically just creeped up until it was above a threshold, and then boom, everything happened. You don't know, because no one's getting monitored on a daily basis for their entire life so that you can find this. But with what we're learning from trauma is that actually you can potentially utilize the mechanism that you infer from that and just play them out longer over time. We could run these simulations for 10 years and you absolutely will have this self-sustaining inflammation or chronic inflammation, to me, is the chronic restarting of acute inflammation. That cycle that I showed you in SARS-CoV-2 is immunology. It is not just virus infection. What you have is you have a stimulus, you have probably some amount of innate immune response if it's an infection stimulus, but you really have this damp-driven stimulus for secondary inflammation, which activates adaptive immune circuits, which have slower on but also slower off kind of rates, which means that you now can set in motion a vicious positive cycle that can take place. That will have ebbs and flows as you drop below a threshold, which is a characteristic of every autoimmune disease. Add to that, often it's triggered by stress. Stress is just another word of saying neuroimmune activation. And that leads to that restarting and that you're up again, right? So the model that we have of SARS-CoV-2, which I made the connection to all viral infections. Really, it's a model of all immunity. It's the entire immune system, simplified to 4 variables. So because the insights from it are exactly the same, right? Just the number of knobs that you can tweak therapeutically is not as big as it would be if the model was bigger, but that's fine. You can always expand the model. But if you start by trying to think of, let's make the biggest possible model, that's a mistake. You really want to start with the core engine being the right one, get it to work, then expand outward. Make your foundation solid first, then expand to the 3rd and 4th and 100th floor. Don't start from floor number 55 because, the building might not be. I suppose that's a different branch of engineering, I guess. But
yeah. So I think, Tachi, I don't know if you have anything else that you would like to add, or maybe my colleagues or students here. If not, we would probably let Yoram go. And thank you so much again for your time. It's been a pleasure, really. And I'm glad that you accepted this invitation. Everybody has been excited here. Okay, on a side note, I'm not sure I will attend the meeting tomorrow because of teaching, but I will try to. We'll keep in touch anyway. And who knows, maybe someday we can have you here in person. That would be.
I hope next time with a little more lead time we can plan around it, and I'd be delighted to be able to come and interact with people more personally.
Sure. Okay. Thank you very much. Thank you all of you.
All right.
Thank you very much. Bye-bye.